It's no secret that the environment is changing due to average increase in temperatures and other factors caused by human influences. These rapid changes in the environment impact ecosystems, which in turn impacts the plants and animals within. Without these organisms, the ecosystems would not function as efficiently and may even collapse. One example are the plants in the marsh that not only live there, but contribute to the structure of the marsh. However, populations of these organisms can change over time through evolutionary processes. This means their responses to persistent environmental pressures will not always be the same. Coastal salt marshes are just one example of ecosystems that are threatened due to climate change. The stability of the marsh depends on organisms like Chenoplectus americanus, a dominant foundational sedge species found across the eastern coast of the United States. In addition to natural stressors, S. americanus and other important plant species endure the pressures of climate change, such as high salinity, CO2, and sea level, and which in turn impact other processes. The plants must respond to these pressures in some way, but may not always be able to keep up with the magnitude of changes we impose. This leads to environmental devastation, such as transitions from marshland to mudflats and eventually areas of open water. Our lab at Notre Dame is interested in making predictions of coastal salt marsh persistence in the face of global change over the next 100 years. My experiment over the summer at Indy Leaf is one example of how we are studying Chenoplectus americanus responses to global change. I ran a common garden experiment in which I took plants from different locations across the marsh that differed in environmental factors and plant traits. I then transported them back here to Indy Leaf in South Bend, where I grew them all together under the same environmental conditions. I then took weekly measurements of height and stem widths of the samples to be compared across locations that they were collected from. Growing individuals collected from different locations in a common environment will help rule out variable environmental factors that could explain trait differences observed in the field. This will help us understand the breadth of phenotypic diversity that exists across the marsh. When I measure these traits, if I am able to find differences between sites my samples came from, then we can be confident these differences in traits are due to differences in the plants themselves, since the environmental differences were being controlled for. Once we have a good idea of the genetic diversity within this species of sedge across the Chesapeake Bay, we can contemplate the variation of responses to environmental change factors like CO2, salinity, nitrogen, and sea level. But only knowing how the plants of today's marshes respond to environmental pressures isn't enough to make accurate predictions of marsh stability in the future. Since we know that populations of organisms change over time, and therefore so do their responses, we cannot simply assume that the responses of future populations will be the same as the responses of populations now. But how can we incorporate this change into the statistical models we are using to make these predictions? To understand the changes between old and new populations, we need data on old populations. Luckily, the McLachlan Lab has a unique study system we use to essentially go back in time and look at how these plants have been changing over the past 100 years of climate change. The S. americanus seeds can remain dormant for many years at a time, and since the marsh builds up in layers over time, we can extract these seeds from deeper layers of sediment, grow them in our greenhouse to create older populations, and compare traits of these older populations to populations in today's marshes. Understanding how these plants have changed up until now and how they are currently responding will help us to make better predictions of what will happen to the marsh as the rate of climate change continues to increase. Ultimately, we want to know whether or not the marsh will be able to withstand pressures like sea level rise, high salinity, and CO2. Maybe this nifty poem will help bring all of this together. Dr. McLachlan discovered something with S. americana's seeds. He worked hard in the lab with help from his colleagues. They found that after decades, these seeds remain viable, allowing them to germinate century-old seeds with methods very reliable. Our team takes soil cores from the marshes of Chesapeake Bay and dates them radioactively from 1900 to today. For each layer of soil, a date was then estimated so that the ages of the seeds could then be calculated. Layer by layer, these aged seeds were isolated, then washed and rinsed and prepped to be germinated. After decades of uninterrupted sleep, we ended up with resurrected plants we could keep. The plants were cloned out to provide old populations that are now used for evolutionary demonstrations. We grow these old plants among modern ones in our experiments so that we can make inferences on trait inheritance. We subject the plants to different environmental conditions, such as high sea level, salinity, and CO2 emissions. These experiments address questions about plant adaptation to environmental changes that threaten the nation. Marsh ecosystems are just one of many things at risk. In the face of sea level rise, will the marshes persist? Collaborators at Smithsonian Environmental Research Center want to know just how much environmental pressure these plants can forego. Because the fate of the marsh depends on these organisms, who provide immense biomass through crucial mechanisms. An ecosystem process known as accretion will protect these marshes from forcing deletion. As the sea level rapidly continues to rise, the elevation of the marsh helplessly complies. But if the rate of accretion cannot be maintained, areas of open water will be all that remain. This jeopardizes the marsh's valuable ecosystem services, such as habitats for animals and protection against storm surges. So through our efforts, and those of whom we collaborate, we are on a mission to question, hypothesize, and educate. We will incorporate the data we have from phenotypic differences into a statistical model that we can use to make inferences. Data can also be used by environmental regulators and even inform the decisions of our legislators. With our aim in determining evolution's role in the marsh's fate, we act as urgent and efficiently as we can before it's too late. For more information on the Coastal Marsh Ecosystem Dynamics Project and other research in the McLachlan Lab, visit the McLachlan webpage. Thank you for listening.